Today's topic is Meristem and shoot tip culture. Morel and Martin firstly developed the technique of micropropagation on the dahlia plant, and the motto of this micropropagation was to develop virus-free plant. Means for the eradication of virus, Morel and Martin developed the technique of micropropagation. This technique, this was done in 1954, and after 10 to 11 years, Morel in 1965 again done this micropropagation on an orchid cymbidium. And the technique of micropropagation gained momentum in 1974 when Morel and Murashik mainly emphasize on the three stages of micropropagation. It aware the other scientists about three different kind of medias are required in different stages. Not a single media is sufficient for the complete regeneration of the whole plant. And this technique of micropropagation is more successful in herbaceous plant. Why so? It is more beneficial for the herbaceous plant is that herbaceous plants has weak apical dominance, so it will not only increase in size, it will also develop axillary shoot. And when we do meristem or shoot tip culture, we take the tip portion of the plant, okay? And this tip is a dome-like structure. This is a dome-like structure, the tip of the shoot. This is apical, apex, and apical meristem is a dome-shaped structure. And in here 2-3 young leaf primordias are also present at the apex portion. So apical meristem with these leaf primordias are called shoot apex. Okay, apical meristem with few leaf primordias are called shoot apex. Now the first stage is isolation or selection of explant. Selection of explant means from which plant we want to take explant and what is the exact part of that plant. Either we should take leaf, root, stem. What is the explant we have to decide, and what kind of the plant, and what is our motto of the micropropagation. On the basis of this we select our explant. After I'm discussing the stage 1 selection of explant. When we have decided we will take the explant of this plant, what we do, we take a small portion by which we will continue our regeneration process. And after we have take out the explant, what will needed, now we have to do the sterilization of that explant, surface sterilization. For this, various type of chemicals are available in our laboratory. It can be T-pol, twin 20 kind of the chemicals by which we sterilize the explant. And then what to do, established on the nutrition media. Student, here nutrition media used is generally MS media. Okay, MS media is used in the first stage, and MS except MS, other medias are White's media, Gottfried media are used in the first stage. Student, what happens in the first stage? Here shoot formation also take place. So what is the need? Here need is to supply cytokinin, because cytokinin helps in the shoot formation. Okay, and the best cytokinin here, 2-3 kind of the cytokinin are used, like benzyl adenine, kinetin, 2-IP are used in the first stage where shoot formation will take place. And after this, when here one shoot is formed, now we will enter in stage 2. What happened in stage 2? Here proliferation of shoots take place. Means in the stage 1, which shoot was increasing in size, now developed axillary branches or axillary shoot formation also take place in stage 2. This is the stage 1 and this is stage 2. As we have discussed, for the shoot formation, the apical dominance should be lower down. Student, one thing always remember, not only in stage 1 and stage 2, shoot formation helps not only occurs not only with cytokinin, but here small amount of oxygen also needed, and the cytokinin and oxygen ratio for the shoot formation is 10:1, and here axillary shoot formation take place. And in the stage 2 also the cytokinin required are benzyl adenine, kinetin and 2-IP, but difference here, high concentration will be used. Comparative to stage 1, there was low concentration, but in stage 2, high concentration of cytokinin will be used. And next stage is stage 3. What happens in stage 3? In stage 3, we transfer these shoots, it can be either a single or multiple shoots, to the rooting medium. What is rooting medium? Which hormone I tell you in the earlier lecture? The rooting hormone is auxin, but auxin do not work alone. It helps, it promote the formation of root with cytokinin, and its ratio should also be 10:1. If we give only auxin in high concentration, what will happen? Instead of root formation, here callus formation will take place, and here auxin and cytokinin ratio will be 10:1 for the root formation. Okay, and here the auxins used are NAA, IBA, IAA and 2,4-D. Here auxin will also used, I told you, in stage 1 and stage 2, but it should be weaker auxin, means 2,4-D should not be present as it is strong auxin. And when rooting is formed, rooting has developed, means a plantlet is formed. Now this plantlet is transferred to a pot to give its exposure to the natural field, natural soil, and then plantlet will develop the complete plant, and then plantlet is developed in the soil after pot. And this is micro propagation. Students, one important point left is that many plants are rich in polyphenol. Last point, this is very important to tell you, is that plants are rich in polyphenolic compound. And what happens when we isolate the X plant? There are chances of the injury in X plant. So some oxidases, polyphenolic oxidases, which are present in environment, what they do its oxidation, and by the oxidation the tissue turn blue, black, and ultimately the tissue is killed. So how this problem is resolved? This can be done with the help of some antioxidants are added in the nutrition media. What kind of the antioxidant? They can be ascorbic acid or citric acid. We can add directly to the nutrition media, or we can give its pre-treatment with these antioxidant before adding to the nutrition media. And third, when we take isoplant or explant, or in initial period of its micropropagation stages, we give it's in a reduced light or in a dark area, because there are more chances of oxidation in the presence of the light. And fourth is continuously change the nutrition media, so that there will be very much less chances of the oxidation. So this is complete.